Hello everyone, my name is Ashfaq Ahmed, and today we will talk about exploring research ideas in bioinformatics and computational biology. This is a new video series, and this is the first lecture of this series. We will cover too many things from tutorials and different methodologies in this series, but today is the first video of this series, and um, basically I will cover the primary concept of bioinformatics research and computational biology research if you are interested in. So this video is very important for the research students or those students who are interested in any kind of research, and specifically protein research. So let's start. What you can expect from this talk. In this talk, I will deliver a basic concept for protein and its connection with research, and how to start a research project related to protein, and what to care and what to target, and these three basic stuff you will actually learn. So this is a simple slide, and here I will tell you what are protein structures and how they are formed. So if you can see, here I have shown you a cell, and the cell has a DNA, and the DNA is transcribed or translated into amino acid sequences. What are amino acid sequence? It's just simply a linear stretch of amino acid connected by peptide bond, and these sequences are further folded into a unique structure, and that unique structure that contains three elements, to which we call secondary structure elements. These elements includes alpha helices. You can see these are alpha helices, beta sheets, the sheeted like structure, and some loops. The loops, normally we call it connections between these secondary structure elements. They looks like threads. So go further. Okay, this is a typical structure of amino acid. So remember, all the amino acid, they contain a similar structure, all the 20 amino acid except this side chain, the side chain which is denoted as R or alkyl group, whatever you can say. So upon the generation of primary sequence, you will have this kind of structure, or to what we call it primary sequence of protein, or primary structure of protein also some people call it. So this is the typical primary sequence of protein, and this is a individual amino acid. They are connected by peptide bond, and there comes a condensation reaction which removes water and generates a peptide bond between different amino acids. Okay, so at the end you will have this kind of structure. You can collect this kind of structure from different databases like NCBI, like UniProt, and once you have the primary sequence analysis, then you can do many things, and we will discuss later on in this series that what can you do with this primary sequence of a protein. Okay, so here we will talk simply about the folding. This primary sequence folds into this kind of structure, to what we call it protein folding. So how this structure folds basically to take this kind of structure. So it has many factors included, and all these factors are because of the R group. For example, the charges contained by the R group. For example, if you see this example, the E and D, the glutamic acid and aspartic acid, they has a negative charge, but the H and R, histidine and arginine, they have a positive charge. So by naturally they both contain the opposite charges, so it will be attracted. So you can see here the primary sequence, it will fold some like this way. The positive and negative will come close together and they will make a bond. To this bond we will call it salt bridges or electrostatic interactions. Or okay, another property the side chain has, that is the hydrophobicity or hydrophilicity. If the side chain is of a hydrophobic nature, it means they don't like water, so they all such kinds of amino acid will group together like here. So the hydrophobic will group together, the opposite charges will comes together, and the same charges will stay away or will tend to stay away from each other. So this way a unique folding will form. And finally, if there are some sulfur groups contained by the amino acid which we call it cysteine, and if they are in a close enough closer to each other, so they will form dicysteine bridge or disulfide bridge. So in that case, that will be the third factor. And finally, hydrogen interactions, it will also be formed. You may know, or if you don't know, so you need to know that the hydrogen bonds, in a hydrogen bonds, at one end there will be hydrogen and on the other end there will be oxygen or either nitrogen. So I'm not supposed to cover the primary, secondary, tertiary and quaternary structure of protein in this video, but there are two kinds of hydrogen bonds. They are taking part in protein structure folding. The one we call it intermolecular and the other we call it intramolecular. So collectively, the protein folding takes place because of four main factors. One is the hydrophobicity and hydrophilicity of R group, and the second one is the charge contained by the R group. The third one is the if the presence of disulfide bridges or not, and finally the hydrogen bonds, both intra and intermolecular hydrogen bonds. And at the end of the day, we get a tertiary structure or quaternary structure for some proteins, because not all the proteins contains the quaternary structure. So that is also another issue. We may cover it in some other videos. So finally you will have this kind of structure, and to what we call it also a 3D structure or three-dimensional structure of protein, or a completely folded structure. So that's how it works and that's how it folds. Okay, this slide is about the basic understanding of protein function. So many people, they don't know, or they even, I would say, they don't care to look for these things. By knowing the protein name itself, you can expect or you can guess the function of that protein quickly, I would say. Simply how I will teach you in this slide. Uh, but first of all, you should know that the proteins, they basically perform three kind of functions in the cell or in an organisms. The structural proteins, you have some structural proteins in your body, and they are making structural elements in cell wall and in cell membrane, even in body parts like in nails and in hairs, and in many other stuff. You can google it. So to such kind of proteins we call it structural proteins. But there are catalytic proteins also, and they perform as an enzyme. They catalyze certain reactions, they speed up certain reactions, they slow down certain reactions, they inhibit certain reactions without changing its own self, and we call such kinds of activity, we call them enzymes. Okay, the enzymes, the third category is also, the enzymes are also involved in the third category, but I have removed it into a separate category and I called it regulatory proteins. The regulatory proteins are basically transcriptional factors or transcription factors, and they targets, remember, the transcription factor targets the double-stranded DNA to initiate or to control, or I would say to regulate the transcription process. When I use the word regulate, so it has three meanings: to turn it on, to turn it off, or to make it slow or make it fast, So uh, the regulatory proteins, they are also, most of the time they are enzymes, and they have to regulate the process of transcription. Okay, and thus they need some different level of oligomerization, but you may not know or you may not understand oligomerization at this stage, so skip the oligomerization part now. Okay, coming to this part, in the start of this slide I told you that by knowing the names of a single protein you can guess its function. And actually, and that guess will not be so much wrong. In fact, it will be right. For example, uh, if a protein name has a word kinase, so remember, it will catalyze, or it will assist, or it will help the addition of phosphate ion. It means if you are reading a protein, for example protein A kinase, so without knowing the literature you should know from its name that this protein will perform kinase activity. Now what is kinase activity? In kinase activity, this enzyme will take phosphate ion from somewhere, let's say, I would say from ATP, and it will give it to some another substrate. This is actually a kinase activity. And the second one, for example, if you are reading a name like protein A carboxylase, so it actually catalyzes the removal of carboxyl group. So a carboxylase will always remove the carboxyl group from any substrate. Okay. The third, if you are reading like protein A phosphatase, so in this case it is actually phosphatase is the opposite of kinase. The kinase actually aid or catalyze the addition of phosphate ion, and in reverse the phosphatases remove the phosphate ions from the substrate. Dehydrogenase will catalyze the removal of hydrogen ions, and so on. Phosphotransferases is actually the middleman between the kinase and phosphatases. How? Because they will transfer the phosphate ion from one place to another. So uh, in the from this slide, now you know that protein perform catalytic, regulatory, or structural functions. but you can also know from its name its general function. It doesn't mean that you always need too many papers or too many books to know the basic function of a protein. So you can guess it from its name. Okay, so uh, this is a very important slide. I have picked it from somewhere. So protein, basically, how many function a protein performs. So there are many functions, but if you group them into a single place, so there are totally 6 functions the protein will perform. For example, the first one is regulation. It will off or it will on a certain process, or it will slow down or it will fasten a certain process. So we will call them in a scientific term, we will call such proteins regulatory protein. Okay, protein will also perform in structure, like I gave you an example before. Okay, protein will also perform in movements. In muscles, there are too many proteins and they are involved, and even the biggest gene of the human body is DMD, dystrophin muscular dystrophy gene. So technically you can say the biggest protein. It contains almost 3600 amino acids. So and a very popular muscular disease, Duchenne muscular dystrophy, is linked to this protein. So actually I wanted to convey that proteins are also involved in movements. And catalysis to speed up a reaction or to slow down a reaction, to inhibit some process. This all comes in a catalysis. Okay, to add something to some substrate or to remove something to some substrate, uh, this all comes in a catalysis. uh, Finally, in transport, there are many proteins. They perform function as a transporter, I would say. They call it transporter, and these transporters are most of the time they are located on the plasma membrane, or I would say in the cell membrane, and they transport different ions, different stuffs. They import to inside and they also export it to outside. Okay, and these proteins are also involved in signaling. uh, um, they signal different things to each other to perform the normal function of the body. Okay, so we discussed these 6 functions collectively the protein will perform. Now then the question comes, how protein will perform these function. This is important. Okay, you know that it will perform regulation, you know it will perform movement, catalysis, signaling, whatever, but how they will perform these function, this is an important question. So protein will perform these function by these 5 methods. They can perform this function by performing all these 5 methods or these 5 processes, or they can just choose one of them. So what are these processes? Oligomerization, or we call them oligomeric state. The second one is post-translational modifications, or PTMs, or the third one I would say cofactor, which are normally the divalent ions or transition metals like calcium, magnesium, zinc, iron, etc. And the fourth one is PPI, or protein-protein interaction, and the fifth one is conformational changes. Okay, So these are actually the five methods by which a protein will perform certain functions. What functions? These functions. Okay. So I will discuss briefly and quickly these five methods to that you understand how protein works or how the protein functions. So the first one is the oligomerization. For example, this is a this is a basically a regulatory protein, and actually it's a TF, transcription factor. So transcription factors are those proteins which are involved in the process of transcription. The 4R22 is actually the PDB ID. So if you want to have this protein, if you want to see this protein, just open the PDB and type this one, you will have this protein. So if you see, this protein has two copies. This is one copy, I boxed it, and this is another copy, I boxed it. So why this transcription factor needs to copy, this is another question. Because DNA is a double stranded, and if it has to interact the DNA for certain process, it has to interact both the strands. Because you know that the DNA is not only a double stranded, but it is also same and anti-parallel. So if a gene, if certain sequences are present here on this strand here, this is a certain sequence, and if this protein has to interact, these sequences, so the same sequences will be here. So sorry, here. Okay, so the protein has to interact these sequences in both places, here and here, and for the reason it has to generate a dimer. And if you see, these two parts are mirror images, they are exactly similar, and we call it dimer. To one we call it monomer, and if there are two we call it dimer. So such kinds of processes where a protein recruits its another copy, we call it oligomerization. Okay, and why it recruits another copy? Because it is the need for the function. As I told you, in case of DNA, the similar sequence is also here, and the similar sequence is also here in this chain. So if for example, if this protein has to inhibit this sequence, if let's say this sequence is a gene, for example, this protein has to inhibit the function of this gene by binding. So the protein needs to bind both copies. In that way, the protein needs two copies to bind both sequences located on different strands of the DNA. So in this case, we will call it that the protein has a dimer. For example, for certain function, the protein may need three copies. So in that case, we will call it a trimer. For example, the famous protein, spike protein of the coronavirus, which attaches the receptor located on the in the human system, ACE2. So the spike protein normally attacked or interact the ACE2 receptor in trimer that recruits three copies. So actually this process of recruiting its own copies to complete certain function, we will call it oligomerization. And if it recruits two copies, so dimer, three copies trimer, four copies tetramer, and so on. Okay, in B panel, this is a transporter, and I told you in early slide that transporters are normally located on the membrane, So this is actually a transporter, and the transporter, they also most of the time they worked as a dimer. You can see here one monomer, and this is another monomer separated by a different color. Why here they need a dimer? Because they import or export by the conformational changes, but I will not go in so much detail for transporter here. Okay, so um, we talked about the cofactor or zinc transition metals. So in this slide you can see uh, this is the part. You can see this is the cytosol part. This is actually the plasma membrane. You can see IM stands for inner membrane, the center stands for periplasm, and the OM stands for outer membrane. This portion and the extracellular matrix, this one is the outside part. So if you see, it's a very beautiful figure I have selected. This is the iron part, this is the zinc and manganese part, this is the copper part, and this column is the nickel and cobalt part. So if you can see, so many proteins, they require iron for transportation or for different functions. Even in case of iron, the classical example is hemoglobin working in your body. So without iron, there will not be a hemoglobin. Okay, the same way zinc and manganese, copper and nickel and cobalt. So uh, that actually, this slide actually is telling you that how much important these transition metals are, these metals for protein functions are. If they are missing, the protein will not perform its function. So this is a special case. I want to discuss it quickly. We have four structure: carbapenem, manobactam, penicillin, and cephalosporin. These are basically four classes of the beta-lactam antibiotics. All of them contains beta-lactam ring except this one. That's why that's called manobactam. A half ring. It contains almost kind of half ring. Okay, so if you see these two zinc ions, they attacked on this beta-lactam ring, uh, and we call it amide linkage, and eventually they break it. And once this this is actually cephalosporin. Once it is broken down, so the 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 antibiotic will not be able to perform normally, or I would say the antibiotic will be hydrolyzed and it will be inactivated. So that way a person will have antibiotic resistance because the antibiotics he is taking is not functioning properly, or it's hydrolyzed. Okay, and where these zinc they where they they are present to attack such kinds of beta lactam rings. So they are present in a protein, we call it beta lactamases, and these two zinc ions are present in beta lactamases, and they attacked on such kinds of like here I showed you oxacillin. These two zinc enzymes, they are present in a protein. Okay, and third one is conformational changes. I'm going a bit quicker now because I think the video is getting longer. So what are conformational changes? See this video. Did you see uh, a protein moves, this part moves from down to up, and as a result it has captured this yellow part. Okay, So this kind of change where coordinates the protein or the amino acid atoms has changed its coordinates. So in this kind of change we call it conformational changes. So a protein change the conformation to attain certain functions. Most of the time they stay active in one stage. For example, this is active stage and this is inactive stage, the previous one. So to attain certain function they change its conformation and become active, and once the process is completed they goes back and become inactive. Okay, this is another example. We call it shear movement. So this kind of movement we call it shear movement. It's not a complete conformational change, but it is a kind of smaller movement, and we in science or scientifically such movements are called shear movements. Okay, let's move to another slide. This is a complete conformational movement where the domain perform angular rotation. You see again. So this was the part 3. Before we have discussed that proteins perform these functions, these 6 functions by oligomeric state we have covered, by cofactor or transitions, by methods we have covered, by conformational changes we have covered. Now we will cover about the protein-protein interaction. So in this figure you can see that a simple protein, I have developed this figure from the string database. So let's say this is the GRAS. It's interacting almost 10 proteins, but normally our students they will show us such kind of figure generated by string database, but you don't need to just tell people these kind of figures. You have to work further on it. What kind of work you can do on it? For example, are these 10 proteins interact at the same time? It's a question. Might be not. This is the answer. Or some event recruited this protein, or this how this protein comes here. Maybe because of this, maybe because of this, that it interacts this protein and it is recruited here to interact with GRAS. Okay, let's be specific. When GRAR interacts, and to what interface? For example, this GRAR interacts here. So which interface it is using? Interface is the location used for the interactions. Protein do not interact blindly. They are very, very, very specific in interaction. So what interface they are using? For example, they are using this interface, this side, to interact here. So this protein also interact this one, VRAG. So what interface it is used here? If the interface is same for GRAS and VRAG, it means that at the same time they do not interact with these two proteins. So the interface detection is very, very important. How many interaction interfaces are located in GRAS? On this protein, how many interaction interfaces are located? For example, 3. It means at the same time it will interact 3, not all the 10. So for what functions to perform GRAR interacts with GRAS? Why this protein interact to this protein? So before these were generally certain questions I generated for you, that showing or just picking such kind of figures will not work. You have to further work slightly more on this, and to dig out and to strengthen your manuscript and your research. Okay, So conclusion, you need to be always knowledgeable, you need to be always specific. Do not grab each and everything and put in your thesis or in your manuscript. And also be scientific. It means that do not work just haphazardly, work with certain questions and work with certain methodologies. Okay, to so uh, we will cover in next video that what how you can start a research project, or what you can extract from a literature study, and that video will be the continuation of this video. So you need to understand all these things, and see you again. If you have any question, drop in comments, and I hope you will enjoy this video. Stay connected. Bye bye.